Many of the early ideas in BioCAD at the start of synthetic biology grew out of systems biology with a heavy emphasis on simulation. These types of tools were central to both fields. In many ways, synthetic biology has become its own clade of forward design tools that only partially overlaps the system biology tool space around biochemical ontologies and these simulation tools. The basic idea of a simulation is to define an initial concentration of each molecule type in the cell as a variable. You then write out equations that describe how these concentrations will change as a function of time, as a function of the concentrations of the other chemicals. You then simulate what the system will do either by numerical solutions to differential equations or a stochastic simulation. Often computational biologists use these simulations to model the behavior of complex biological systems. In the context of synthetic biology, these tools allow the engineer to model different circuit architectures to decide which ones might be better than others. Here's an example of a model from Ron Weiss. Shown is a cartoon of the process they wish to model. The pulse generator cells encode a constitutively expressed LuxR gene, which produces the LuxR protein. If LuxR binds to exogenously provided homoserine lactone, it will activate LuxI promoters. Activation of these promoters leads to the production of GFP and C1 protein. Once C1 accumulates, it will shut down production of GFP, resulting in a temporal pulse of GFP expression. Computationally, they describe this system in terms of 23 species doing 44 reactions. Here we show just 3 of the reactions. Each term describes the concentration of a different chemical species in the system. For example, these reactions all describe transcriptional, translational, or decay processes. Similar models can be written for enzymatic transformations. You can then translate the system of chemical equations into a series of differential equations. Here we describe how the concentration of RNA-P changes as a function of time in a manner related to the concentration of an mRNA and its decay. You can then adjust the parameters in these models, which encode information such as the translation or transcription rate. By examining how the model changes in response to these adjusted parameters, you can examine the scope of behaviors that this particular gene regulatory network is likely to exhibit. In this case, they are able to fairly precisely describe the observed pulse behavior for the circuit they built in the wet lab. Often stochastic and differential equation-based modeling is done in MATLAB or in GUI-less C programs. There's nothing to look at. However, there are also specific tools for modeling and illustrating the results of biochemical network simulations. Many such tools exist within the domain of systems biology, and a few carry over to synthetic biology. TinkerCell, developed by Herbert Soro and Deepak Chandran, provides a user interface for drawing genetic circuits in the same way that lab researchers typically draw them on paper. The software translates this architecture into an SBML model, which can be translated into a differential equation or stochastic model and then simulated.